Sliding filament theory. Muscle contraction is the process where muscle fibers generate tension to produce movement or maintain posture. It involves a complex interplay of electrical and chemical events, primarily described by the sliding filament theory. An action potential triggers the release of calcium ions, which bind to troponin, causing tropomyosin to shift and expose myosin binding sites on actin. Myosin heads then attach to actin, forming cross bridges and pull actin filaments toward the center of the sarcomere, shortening the muscle. ATP provides the energy for myosin to detach and reattach, continuing the cycle until the signal stops and the muscle relaxes. Structure of muscle cells. Muscle fibers. These are the individual muscle cells that are long and cylindrical. Each fiber is multinucleated and contains specialized structures for contraction. Myofibrils. Inside muscle fibers are myofibrils, which are long, rod-like structures composed of repeating units called sarcomeres. Myofibrils run the length of the muscle fiber and are responsible for muscle contraction. Filaments. Within each myofibril are thin, thick filaments. Thin filaments are primarily made of actin, while thick filaments are made of myosin. These filaments are arranged in specific pattern within the sarcomere, allowing them to slide past each other during muscle contraction. Actin is the main protein in thin filaments with binding sites for myosin. Myosin is the main protein in thick filaments using ATP. Nerve signals trigger calcium release, exposing binding sites on actin. Myosin binds to actin, forming a cross bridge and pulls the actin. ATP helps myosin release and reset, repeating the cycle to shorten the muscle. Calcium triggers the muscle contraction by exposing binding sites on actin.